Okay, so without any further delay, so I'll be talking to you about a normal immune system. It is important to understand what Prof. Chung is going to talk about later. So it is important that you understand how your normal immune system works. Many of you might have had some information about this or read about it somewhere. So I'll just reiterate that if I will. And then how does immune system play a role in myeloma, and how are we trying to harness it? And I have a very good interesting video at the end of it. So bear with me as I take you through this picture. Now, our immune system is very complex, hugely complicated, and that is why it probably has taken us better part of one whole century to try and make some sense of it. So the cells on the left side of this is what we call as innate immune system. Innate immune system, you can use another name. If I can use another name, it is just something that you're born with, something that is, probably a little bit crude, not that specific, but you have it within you. Adaptive immune system, on the other hand, is something that you acquire. By example, by being exposed to viruses, germs, whatever foreign things that come into you, the arm of immune system that deals with it is the adaptive immune system. By and large, the immune system is being controlled by a lot of, or being orchestrated by a lot of white cells, and there are white cells come in many different shapes and sizes. You might have heard about neutrophils that we talk about often at your appointments in clinic. You're neutropenic, which means that you're low in neutrophil counts and so on. But there are more important cells as well in your immune system that comes on the right side of this adaptive immune system. Now, neutrophils, macrophages that form part of the innate immune system is like, I would usually tell my patients, they are like police. They don't have very sophisticated weapons, but all they do is just they find problem, they go and attack and try and solve and diffuse it. That is all they do most of the time. But this is how wars were fought back in the 1800s, right? It's not enough anymore. We have more sophisticated germs, more sophisticated infections and cancers, and hence we need to get our immune system up to speed as well, and that is where the adaptive immune system comes in. That is a bit like intelligence. So we need to gather information, we need to be targeted, we can't just go blindly and kill everything and anything that comes in our path. So we need to have missiles, we need to have satellites, we need to have spies even. So that's how the adaptive immune system works. Two main cells that we come across in the adaptive immune system is B cells and T cells. There are many, many more. I'll primarily be talking or paying attention to these things. They have many weapons. One of them is the antibodies, which are very specific targeted proteins that are being produced by plasma cells, which, as you all know, is the cancerous cell in myeloma patients. Then we have T cells. Now, much of information about T cells was not available till about in the last 30, 40 years. So we thought that maybe T cells have a function, but we didn't know what exactly they did, and especially how they worked. Not much was known. Now, in terms of the anatomy, how things are organized in the immune system is via a network of what we call as lymphatics, which is just basically very tiny kind of blood vessels, if you will. And they help to transport these immune cells from one part of your body to another. Of course, we've got a lot of area in our body that needs to be always monitored. We have about more than 1 meter square of skin where germs and foreign things can enter. We have much longer meter square of bowel, for example, that needs to be monitored, and we always are exposed to these foreign things, and we never know when one of these germs are going to enter into our body and cause problems. So our immune system has to be all-pervasive and has to be everywhere, really, and that is how exactly it is organized. So you have lymph nodes interconnected by lymphatics, you have spleen, you have liver, you have bowel, everywhere the immune system is there, is present. It's kind of omnipresent. Now, this is the story part, so that's a little bit of science that I'm going to talk about. So story is mostly about immunotherapy. Now, this is a book that I read as a medical student back in the day, seems like a long time ago now. The title of the book, I don't know if you can see at the back row, it's called Commotion in the Blood, Life, Death, and the Immune System. The author of this book is called Stephen Hall. So the story is about how the immunotherapy, as we call it now, evolved. So back in the day, in the 1800s, in 1890 to be specific, there was this young, beautiful girl who lived in New Jersey called Bessie Dashiell. Now, she was friends with one of the most wealthiest American families, the Rockefeller family. Suddenly one day, Bessie gets severe wrist pain. And lo and behold, it turned out to be cancer. To be specific, it was a type of soft tissue cancer called sarcoma. Not much was known about it, obviously. It was over nearly 150 years ago. And these were the days when even a lot of information on how to diagnose cancer was not available. So the treatments were rather crude. What did they used to do? Well, they usually used to go to see surgeons who chopped off the part that had the cancer. Unfortunately, that was done for her, and the surgeon took out the whole arm because obviously he believed that is the only way to get rid of the cancer, and that was the day of treatment for sarcomas in those days. Unfortunately, what they didn't know, they didn't have scans, they didn't have anything at the time, and hence they didn't know that the cancer already spread far and wide, and Bessie died within a very short time after the operation. Now, because they were very close friends with the Rockefeller family, John Rockefeller was really affected by Bessie's death, as is often a problem when we lose a very young or an adolescent patient to cancer. And it really touched her treating surgeon, who was William Coley. Now we call him as a father of immunotherapy, but it really started off with this patient. So William Coley thought, how really Bessie's death affected him deeply. So he went back and thought, how can we change the approach to cancer? We can't just keep chopping off parts of bodies which have cancer. That doesn't make any sense. So he went back to the hospital records, and the hospital that he was working at is called Memorial Hospital, which we now know is the Memorial Sloan Kettering in New York. And he identified one particular patient, Mr. Stein, and what struck him was, despite the disease seems to be exactly the same as Bessie's, Mr. Stein survived. And when he compared the cases, what he found was different with Mr. Stein was when he had the time of surgery, he had a severe infection. So they thought, maybe there is something going on here. Why if we create an infection artificially to patients with cancer, by introducing something into them, will it work? Was a question that he wanted to answer. Now, much of the subsequent things that he did will not even get through the first step of our scientific rigor these days. They were not ethical, and it wouldn't happen today. But that is how it started. So what he tried to do was introduce infections to patients with sarcomas, telling them that this is what is going to happen. One particular case that struck, and which he reported later in the literature, was a case of Mr. Zola. Now, Mr. Zola had a very similar sarcoma like Bessie in his throat, of all places, which was nearly stopping him from breathing. And Dr. Coley introduced the same kind of infection that he used for other patients with him. Now, many times this kind of treatment worked, but many times it didn't. So he didn't really know or have an answer as to why it works for some patients and why it didn't work for other patients. But in his case, he didn't give up, and thankfully so. He went for a stronger and a stronger bacteria, and eventually he found one bacteria that elicited an immune system response in Mr. Zola, such that the sarcoma completely vanished. And he lived subsequently for many years to come. So that he considered was an important discovery. However, it has to live through the establishment. The medical establishment in those days was epitomized by none other than Dr. Ewing. Now, Dr. Ewing has a different type of bone cancer named after him. He was a fantastic researcher in himself. And he thought, what crude thing is this? How do you even fly a treatment like this, artificially introducing infections into patients? It just doesn't sound right. So there was much of Dr. Coley's life was very, very tough. He couldn't get this through what he thought was genuine life-saving treatment for patients, but it didn't fly. Now, Fast forward, so that was back in 1920s. Now, if we fast forward it to 2010, we shall come to why the year 2010 is important, but much of this 70 to 80 years has been invested world around by doing basic biomedical research to try and understand immune system. What are the arms of immune system? What are T cells? T cells, the T stands for thymus, which is a gland that is right there behind your breastbone, which kind of educates these type of white cells to identify what is yours, what is self, and what is outside, what is foreign. Similarly, B cells, the B stands for something else, but we can say they are bone marrow, educated cells. So much of this understanding has all gone and been learned in the last 50, 60 years. At that time in 1920s and 30s, when this was all going on, slowly the cancer treatments evolved. They, involved surgery, which was fine-tuned such that, it wasn't as crude as it used to be anymore. Radiation therapy came into being, and radiation therapy was one of the things that Dr. Ewing was doing, and he clearly showed why his treatment was going to be superior and more or less crude than what Dr. Coley was trying to do. And obviously, chemotherapy came around after the World War II, and that really took up and improved survivals, although it was quite toxic in itself in those days, the chemotherapies that they were using. But however, people's interest grew in all these kind of fields rather than immunotherapy, which was kind of lost as a treatment modality. However, simultaneously, research was being done to identify and learn more about this. Now, T cells in particular, in the last several decades, have come to be known as some of the most robust killing machines, if you will, for tackling cancer. But still, people did not know how they identify cancer cells versus non-cancerous cells, because if you destroy everything, it doesn't serve the purpose. It's a bit like going back to taking the whole arm off for a surgery for cancer. But advancements were slowly made, and mechanisms were found as to how they identify the cancer cells and how they can tell apart cancer from non-cancer cells. And more importantly, how do we harness this power of the T cells to be able to control the cancer cells? Now, if we go back again 30, 40 years, we may not have realized this when the first treatments evolved, but stem cell transplant is one of the most effective forms of immunotherapy, especially allergenic stem cell transplant, where you receive a stem cell from a donor, maybe your own brothers and sisters, or sometimes from an unrelated donor. Initially they thought, it was the chemotherapy that worked for these patients, and hence the disease got cured. But then subsequently it was very quickly found out that it is actually the T cells that go from the donor to the patient that actually goes and attacks the tumor cells, and found that was what was actually working. Now, there are several chemicals that these cells release constantly. We didn't know much about their functions and stuff initially, but then IL-2, which is one of the compounds that these T cells produce, just by giving that drug as a drug, when they started synthesizing it, nearly, it might be very low percentages, but 15% of melanoma patients got cured. So that was, a dawn, if you like, for identifying chemicals which can then harness the power of these T cells to be able to work against cancer. Then came the monoclonal antibodies. Many of you might be on this antibody called daratumumab, which we now know is a very effective treatment for myeloma. And similarly, the very first antibody that was actually produced for tackling cancer was rituximab, where Stanford, which is one of the institutes in US, actually, which is where Dr. Darrell went for a few years, was one of the areas where it really revolutionized the treatment for lymphomas. And we are glad that we have a similar rituximab in daratumumab for myeloma at the moment. Similarly, adoptive immune therapy is where you harness the power of the T cells that works against the myeloma, or for that matter, any cancer, and transferring them from one patient to another, or sometimes from the same patient, but then educating these T cells in the laboratory to adjust them such that they can realize or recognize the cancer cells specifically, and then putting them back into the patients. That is what has changed. Now, 2010 was really a turning point for immunotherapy. Why? Nothing great discovery-wise happened, but that was the first year when we started having results of our clinical trials in immunotherapy for cancers in general. So true enough, the big and mighty, as it were, in the field of medicine, the famous journals started publicizing immunotherapy as a dawn, and we knew at that point that it was a start of a new day for cancer treatments and subsequently in myeloma as well. And even some of the knowledge trickled through to some of the common read weekly magazines, including the Time magazine. This was back about 3 years ago now. I'll see if this video plays. So now, there is a website, a charity-based website in the UK where I used to work called Myeloma UK. You can Google YouTube Myeloma UK. They have plenty of fantastic videos, and I'll just try and play this video for you. It probably makes more sense.
Our immune system is made up of a network of cells and tissues that work together to protect us from germs and foreign particles. It recognizes our own cells, but tries to get rid of anything it doesn't recognize. Each of our cells carries a set of identity tags called antigens that are unique to us. Their role is to stop the immune system from attacking our own body. Anything or anyone else carries a different set of antigens, which the immune system removes before it can cause harm. Different types of white blood cells help make up the immune system. They work together to seek out and destroy anything that is foreign. Neutrophils and macrophages respond very quickly to the first signs of bacteria and viruses by detecting the chemical signals they produce. They remove them by engulfing and breaking them down in a process known as phagocytosis. Other white blood cells, called plasma cells, recognize foreign antigens and react by producing specialized proteins called antibodies. These antibodies lock onto the foreign antigen, flagging it up for destruction by other immune cells. Plasma cells also play an important role in building our immunological memory. This means they remember the same foreign antigen the next time it invades the body. They respond by producing large amounts of the same antibody quickly. Once the invading organism has been removed, chemical signals are released to dampen down the immune response. The immune system also tracks for changes in our body and removes any cells that are abnormal. Cancer cells are abnormal and may carry antigens that would normally be recognized as foreign. However, they also have other properties which stops them from being recognized and destroyed. As our understanding of the immune system has improved, new treatments are being developed which use the immune system to recognize and destroy the myeloma cells. This is known as immunotherapy. Some make use of man-made antibodies called monoclonal antibodies, which recognize antigens unique to myeloma cells. Once flagged, other immune cells can quickly destroy them. Other monoclonal antibodies are made with toxins attached to them. When they lock onto the antigen, the toxin is released, killing the myeloma cell without needing to recruit other immune cells. Cancer vaccines are also being tested. This involves an injection of a small amount of man-made myeloma antigen, which allows the plasma cells to produce antibodies that specifically recognize and destroy the myeloma cells. Other treatments being developed include some which block the signals that normally dampen the immune response, as well as T-cell therapy and virotherapy, both of which are designed to recognize and destroy myeloma cells more effectively. Overall, immunotherapy shows great promise as a treatment for myeloma, offering long-term responses with few side effects.
Okay, you can watch these videos again. Then now there are lots of videos on Myeloma UK website, including if you want to specifically think about a drug that you're on, you want to know more about it, there's all the information there. So please feel free to go and look up. Now, I don't have many more slides to go, which is, I hope, a good news for you. So if you can then classify cancers, there are cancers which stimulate or able to stimulate our immune system very, very well. But there are also other cancers which probably are not going to be that well in their ability to stimulate our immune system. Now, myeloma is kind of in between. It's neither too strong or too weak. It does stimulate the immune system to a degree. So if we think about them, treatments for myeloma, quite a few treatment options have now emerged as immunotherapies. Many of you might be on immunomodulatory drugs that we call, ending with the name IMiDs, called lenalidomide, thalidomide, pomalidomide. So these are all immune stimulating drugs as well. They come under this category. They might be called chemotherapy, but they actually function through their immune system. Monoclonal antibodies we talked about. Now, some of the most exciting things that we are coming up with is something called BiTEs. I don't know who named them. They must have been hungry when they put the name to it. But essentially what BiTEs are the antibodies which help to bring one of those antigens that you saw on the video from that are located on top of T cells and bring them close to the myeloma cells. So if the T cells are wandering around in your body without being able to recognize, this is one way to bring them together. So by bringing the myeloma cells close to a T cell, you're making them kind of eat up or kill the myeloma cells. So that's exciting therapy. And I've just heard about a couple of days ago that Singapore is going to be one of the only, if not one or two countries in Asia that we are going to have access to the BiTE therapy in the very near future, which is always exciting. Now, what Prof. Chung's talk will be is on CAR-T treatment, and I don't want to steal his thunder and I'll let him talk about it more. Thank you for your attention.
But it's very nice to see all of you here. Actually, I just want to make a comment that this is the 9th year of the Myeloma Forum already. And if I look at the audience, there are many of you that were right here 9 years ago, right? So this actually tells us that myeloma patients are really surviving a long time now. Of course, we are not quite there yet because actually over this period of time, we have also lost some of our friends, but I think clearly we are making very important progress because if you look back at 10, 15 years ago, I wouldn't be in the situation where I'll be seeing so many of you that are still with us 9 years and, looking healthy and looking well and, go about your daily business. So this is very positive. And what I'm going to talk here for the next 15 to 20 minutes is further signal of progress, right? So, we are now moving beyond just the usual drug treatment, radiation, to, this whole new area of using our own immune cells to tackle the cancer, in this case, multiple myeloma. So actually, without much coordination, I think the talks have moved very nicely because Dr. Lee actually did highlight some of the very basic aspects of myeloma, the progression, and so on, because we may have new members to this group. And Dr. Mauli has also given the very nice basic background to the immune cells and the immune system and how it actually helps to remove foreign cells in our body. And this leads very nicely into this talk on CAR T cells. Actually, I think most of you in the audience would have heard of CAR T cells. Anyone don't know what, or have not heard of the word CAR T? Okay, so one or two only, right? Why? Because in my clinic now, almost every day that I have clinic, the patients come in and then we go through talking about the treatment and so on. Then at the end, they will say, Doc, what about this CAR T that I hear? Is this could I change to CAR T because I'm hearing that it's curing cancers and so on. So there is a lot of hype, there is a lot of publicity in the lay press as well, on the web, Google and internet. There's a lot of these information on the CAR T, which is why I thought that it would be useful for me to address this topic at this meeting and to highlight what are some of the truths and what are some of the potential myths so that we all could be on the same page. And there is no hype without some truth. So the idea is that CAR T was initially developed most successfully for childhood leukemia. Okay, so you see here a picture of children with leukemia. And why did it reach such high level of success is that using this technology, they show that even in children that has run out of treatment options, so usually these are kids that pretty much, is going to die within a few months because there's no other treatment for their leukemia, that some of them, not all, some of them, when given this CD19 CAR T cells that you see here, this word CD19 is mentioned in many of these papers, could actually be cured of the cancer. So going from a disease, you're not talking about just controlling or giving them another few years, they have no good options already, to a cure, is really something quite remarkable, okay, quite remarkable. But I would like to highlight that this is by no means for all the patients. So all the patients in the same group that has no good treatment to give, that has childhood leukemia, when you treat them, there is a percentage of them that may be cured, and in fact, the percentage is probably less than 50%. But this is quite a remarkable achievement, because usually you wouldn't have anything that could be given for this. And of course, for every publicity campaign, you need to have a poster child. So in this case, it's truly a poster child. This is a kid, right? It is this girl down here that has, initially refractory cancer to now growing into a really beautiful young lady that is going to school, achieving well in school. Every year, in fact, she will put a card that is in front of her. She'll hold this card. This is my 19th birthday, 18th birthday. Every year, she's growing, right? So to show that this treatment is actually curing someone, right? Curing people. And so this received a lot of hype, a lot of publicity. This is like a big new hope in cancer treatment, and will be something that's useful for many patients, right? And so this word CAR T kind of stick in our kind of conscience, right? But CAR T just means a technology. So the CAR T that we use to treat leukemia will be different from the CAR T that we use to treat myeloma. So although the word is the same, just like chemotherapy, right, is one word. But the chemotherapy you use to treat myeloma will be very different from the chemotherapy you use to treat lung cancer, will be very different from the chemotherapy that you use to treat lymphoma, right? So every disease, although the same kind of technology or treatment can be used to treat the condition, it is actually different, okay? So although we are seeing that it's showing tremendous results and long-term benefit, because for the child, she has been disease-free for many, many years now, the same cannot be assumed for every single condition. So I think that is a very important starting point. Every disease is different, although the name may be the same. So I'm just going to go through some of these questions. What is CAR T treatment? Is it approved for myeloma? Just in case someone say that, hey, how come in China we can do this? Across the road in KL, I can go and get CAR T. So is this proper or not proper? I'm not saying anything about whether it's proper or not proper, but I'm just going to tell you that for myeloma later on, that, what's the situation, right? Is it a cure for myeloma, just like what we hope to be and what we saw in leukemia, childhood leukemia? And what are the potential side effects? So is it a safe, very easy, very safe procedure? And what does it entail? So I'll hopefully answer some of these questions and summarize at the end. So in Dr. Mauli's presentation and with the video, it's kind of introduced some of these players already. Antibodies, which are produced by the plasma cells, that actually will bind to protein on the foreign body, which in this case could be the cancer cells or could be bacteria, and it will eradicate the foreign body, right? So on here. And in fact, daratumumab, which many of you are receiving, is a kind of antibody that is manufactured and modified, so not natural from our body, but is manufactured, that actually serve the same function, targeting a protein that is present on the tumor cells called CD38, right? And then she also introduces these T cells that are actually immune cells that we have in our body that also recognizes protein that are expressed on the membrane of foreign body, but usually processed by the cells and usually associated with other immune proteins called like MHCs and so on, right? So it is more complicated because it requires the protein to be bound, but also additional signals and interaction between the T cells and the tumor cells. So it's a complex system, not so direct. Whereas antibodies are more direct. Once it binds, it brings in other cells, immune cells, to actually then eradicate the foreign body. Right? So the body actually employs different system of immunity to remove foreign bodies. It's very effective. That's why we can live and in such a dirty environment. Actually, you see, we are exposed to bacteria, viruses, and so on all the time. The body actually have a very effective system which is very complex and employ different agents to actually eradicate this thing. So actually it's the same for cancer. Why is not everyone developing cancer all, at a young age and so on? It's because also we have an effective immune system to, by and large, for many people, to remove these early cancer cells. So they may detect 1 or 2 abnormal cells, they'll kill it off, right? So it's usually quite effective. But of course, as we age and with some interaction with environment, sometimes this system may fail. When you give it enough chance, sometimes it may fail. And when it fails, just like other important system in protecting our body, the cancer cell may start to grow. Okay, so why does it fail? Is that sometimes, as alluded to in the video, the tumor may actually develop some additional mechanism to shield itself and protect itself from these agents, so they go underground, right? They become invisible to the immune system. And the immune system, although they are circulating, they cannot detect them. Sometimes they develop additional proteins to kind of block this kind of interaction, right? So that although the T cells may recognize the abnormal protein, but these additional signals are unable to be activated because the tumor cells have, again, produced some other mechanism to block the additional signals. So the cancer cells are actually very, very clever. For a cancer cell to survive and cause trouble is actually not an easy thing, which is, again, why not everyone is walking around with a lot of cancer. So by the time the cancer can grow and develop and cause problem, it is actually already a very smart cell, a very smart group of cells. So our immune system have failed, really. So how do we then reharness this very powerful weapon, right? And that is where engineering has to come in, tissue engineering. And that's also the effort of development and technology that is being developed around the biomedical field, right? So we have to modify our immune system to make it active again, to make it effective again. And this is where tissue engineering come in, and so we can engineer these chimeric, which means a fusion. Chimera are actually a fusions of 2 different types of things. In the Greek mythology, it's some chimeric organism that has, I cannot remember whether it's a lion head and some other body or something like that, but it's a chimera, right, made up of 2 different parts. So here we engineer chimeric antigen receptor. How? Because it has this part that recognizes the protein on the tumor cells that comes from the antibody, okay? But the antibody, when it's activated, actually employ quite weak killing immune cells, okay? So let's go back to here. It activates NK cells, monocytes, which are not very powerful killing cells, okay? Whereas T cells are actually very powerful, and they can have a memory, so you will always remember. If the cancer occurs again, it will remember and it will kill it again, right? So it has this long-term memory. So it has these advantages. But the T cell system is not as good as the antibody system because it requires other signal. So it's a more complex system. So here we make the best of both worlds. The direct recognition of protein by the antibody part, link it to the T cell part that is very effective in killing the cell, so that now once I recognize the protein on the cancer cells, I can activate like a T cell function to kill the cell. So it simplifies the process, make it more direct, and make it powerful, and potentially make it lifelong because it has a memory, right? So that is the chimeric antigen receptor, only possible because we can manufacture this and we can express this in the T cells and reactivate and make our T cells that has lost its potency or lost its ability to recognize the tumors in our body to become active again. So the advantage is it does not require the additional signal and is fast. Once it recognizes, it kills the cell. It doesn't require these kind of additional signals and so on. And so this is, for example, a tumor cells, and you see these T cells that are abutting. So what it will do is, in the body, this is what will happen. And with further understanding of the immune system, we can make further modifications to these receptors to make it even more potent, to somehow shut off the signal when it has too much side effects, so that it's not really always lifelong. And because if it's lifelong, once you have side effects, you may not be able to shut it down, and it can become dangerous. So we are able to build in these additional safeguards to either increase its potency or increase our ability to actually fine-tune, when it has side effects. So this is all the advance in the technology that's allowing us to do this. So now we have different generations of CAR-T, but the ones that are in the market, approved, and tested very commonly in patients are these second generations CAR-T. Okay, but I want to highlight that although there are CAR-Ts, they are different, right? They are different signaling. The protein that they target could be different. So for example, the exact treatment that is used for the childhood leukemia may not be relevant to myeloma if that protein, CD19, is really not that useful for myeloma patients, right? So we need to find another protein that is useful for myeloma, right? The CAR-T that's approved and used for childhood leukemia may have different signaling molecules inside, right? 4-1BB, and for the myeloma ones, the different ones in studies and trials, they could be different, right? So this may explain why the potency may be different and the side effects may be different. So again, if you read in a blog and say, oh, this one can cause serious problem going to ICU, brain problem and so on. If it's referring to the childhood leukemia one, it may not be the same side effects that we'll see in the myeloma studies. So again, recognizing that the same treatment may be different, right? So how is it done? It's not straightforward like using a drug that you prescribe in a pharmacy, give, and then you just shoot it in. It's a bit more like your experience going through stem cell transplant, right? So we will need to collect your T cells first. So this is a bit like collecting your stem cells, the exact same experience and procedure. Then the T cells that are collected go into the lab. The cells that are collected go to the lab where we isolate the T cells, using some beads, right? And we activate them. And then we do this engineering. We need to transfer the chimeric antigen into the T cells. And usually this is done using deactivated viruses, okay? And the transduced or the modified T cells need to then be expanded because we need enough large volume to go back into the patient so that it can go and seek out the cancer cells and kill it. And then the reinfusion is exactly like the stem cell reinfusion giving back. So to the patient, the entire procedure is exactly the same as a stem cell transplant, right? You have cells harvested, and then after a while, a few weeks, you have cells infused. What is different is what goes on in the lab, right? The engineering and the manufacturing that goes on in the lab, right? So actually to you, it's the same, but actually what is different is what goes on in the lab. Is it a very simple, safe procedure? Is it as simple as a transplant? I would say that some of the side effects are definitely much more severe than the side effects that we see in the transplant because these are very, very potent treatment. And when you have activated immune cells, just like when you have a fever with an infection, it's because the immune system is active, right? You have fever, you feel achy, tired. So these are what we call cytokine release syndrome. But when you have a fever, it is a very toned down effect. Here we're infusing large number of these cells, and if they're really effective, you can imagine that you can be 10 times sicker than when you have a fever, right? And this can be severe, and it can affect the heart rate, the lungs, and so on. And some patients may have to go to ICU to be monitored closely and to have some additional treatment. So some patients can become very severe in terms of side effects. And because it's very effective in killing cancer cells, you can develop tumor lysis syndrome if you have still got quite a lot of disease. And usually for these treatment at the beginning is given for patients with relapsed disease who have quite a lot of disease, so the tumor lysis can be quite severe. And what happens here is that when the cells die, they release a lot of toxins that actually may damage your kidneys. Okay, so patients may go into renal failure as a result of tumor lysis, right? But all these things, as long as we know that they may happen and the clinical team monitor and recognize them and manage them, on the whole, it should be not a problem, okay? But you may still go through these ups and downs, feeling quite unwell before you get better. Some of the early treatment, particularly in the leukemia, these CAR-T can somehow go into the CNS, and cause some neurological effect, which again is different for the different construct that is used. Okay, so not a standard side effects, but some patients did develop it. But more importantly is a lot of these protein targets are not unique to the cancer cells. So they also exist in normal cells, like B cells and so on. So because the treatment is very effective, all your normal B cells and plasma cells will be also destroyed. And so you may have a long-term problem with your immunity, and you may need long-term replacement of immunoglobulins and so on. So you may have lifelong need to replace what has now become missing. And with that, your immunity should be okay. You should be able to fight infection and so on, but it's a lifelong commitment sometimes to having a monthly injection in a hospital and the cost of it that come with it. So it's by no means a very straightforward, simple treatment, okay? And really, the crux of it is to really identify the correct protein to target, and then you open your whole world out to the different immunotherapy platform, of which CAR-T is one of them. So for myeloma, what is the protein? And the protein is this molecule called BCMA that is expressed on plasma cells, which is the myeloma cells, as well as B cell, right? So we don't really go for CD19, which is used for ALL, because myeloma cells are different cells from the leukemic cells, right? And so in the literature, you'll read a lot on this molecule called BCMA, and that's really the protein that we are targeting now. But still, I think it's not optimal because it is not unique to myeloma only. It is also present in normal B cells, normal plasma cells. So you will get those cells affected as well. So the search actually is still on for a better protein target that may be more unique only to the cancer cells. Then next time our treatment will only affect the cancer cells without affecting the normal cells. So I say that although this is very exciting and we will see some BCMA immunotherapy approved in the next few years, I would also say that this is not the end of the story. This technology will continue to get better. The target will continue to get better as well. And to say that CAR-T treatment in myeloma, people have gone for different targets, as you can see here. CD19, CD138, kappa. These early studies have all failed. Okay, so the word CAR-T is the same. These are CAR-T treatment as well. It doesn't mean that they will all work. Okay, so it's important to drill down to what type of CAR-T they failed, right? Until the group in the NIH did a study using CAR-T that targets BCMA, which is this word here called B cell maturation antigen. You can see it at the top. BCMA, okay? So this is BCMA, CAR-T, chimeric antigen receptor, T cells, right? And you see this remarkable response in some of the patients where before treatment you see so many areas of disease, right? You can see this on a PET scan. These are all areas of disease. 2 weeks after treatment, okay, 2 weeks is very fast. All gone, okay, all gone. And you see that your light chain kappa go down from a high level to almost nothing. So suggesting that this is very, very potent. But you look at the 12 patients you treat, right? You only see response in some. You see PR in the first patient, VGPR in one. This stringent CR1, which is probably the patient example that they show, is only one patient. Okay? So when we think about, is this the savior, we're going to cure myeloma, long way to go yet. Because you can see here, the response is probably about 30%. Okay? Since then, many other groups and companies have been developing this CAR-T. So I show here 4 studies that are most mature, meaning that have recruited the most number of patients as most advanced. They all target BCMA, you can see here, and they have slightly different construct at the back end. The signaling molecules is slightly different. They use different ways to introduce the CAR T into the CAR construct into the T cells, but they're all virus-based. Some are retrovirus, lentivirus. Okay, but again, slight differences. And they all will need some type of treatment, just like your stem cell transplant, you need melphalan, right? For CAR T, they also need to give some sort of chemo to allow these T cells, when they go in, to be accepted by the body so that the body don't reject it, okay? And the dosing you see here is slightly different. Some are using lower doses, some are higher doses, and the schedule is also different. Some are given in 1 day, some are 2 days, right? So there's some variability. When we say most advanced and most patient treated, how many are there? This is 2017, so of course by now it's more. At that time, 6 patients in one trial, 24 patients in another study, 40 in another study, right? So by no means, compared to your daratumumab, your carfilzomib, your pomalidomide, which are treated in thousands of patients, these are very early treatment, right? These are mainly in very refractory patients with 7 lines, some of them 14 previous lines of treatment. So these are really very advanced patients. Most of them would have a transplant before. What is the response like? Overall response rate, 50%, 46%, 77%. The China study that is now, this construct and product is now bought over by Johnson & Johnson, is 100% response rate with quite a high percentage in strict CR, but you also see that this trial is the trial where the patients are more early on in their treatment, only 3 to 4 lines of prior treatment compared to these that are almost twice as advanced, right? So it could be that in the future we need to use this CAR T early on, that will have the effect. But the point to say is that although the response rate is good, we don't know whether they are cured or not, because these are 2017, that means the patients that has treated longest is probably only 3 years, right? So it's a far away to say that these people are cured. Suffice it to say that the treatment is very promising, okay? Because don't forget, these patients probably have no other treatment option already, but still are achieving this level of response. What are the kind of side effects that you see? The most worried are these things called CRS, which I said cytokine release syndrome. The patient may go to ICU. And it's quite high percentage. You can see here 80-something percent, but most of them are low grade, because the grade 3 CRS is actually very few. So this is good because it means that we can manage this without much difficulty. Okay, so on the whole, the CAR T treatment in myeloma looks to be fairly safe, very effective, but the follow-up is still short. So we don't know whether it will be a cure or not. And these trials are still ongoing, so by no means this is approved by any of the agency, FDA, European Medical Agency, is not approved yet. The treatment for leukemia and lymphoma, the CD19, is approved for treatment of leukemia and lymphoma. This one is not, okay, but we'll have to see. So the challenges that we face, I haven't even mentioned that, is the cost. Is the cost of the treatment itself. The leukemia treatment in the US is costing $500,000 one shot. Okay, but they say that it's a cure, so $500,000, you cure you, rather than now you take daratumumab for 6-7 years, it's also $500,000, right? But not everyone is cured. So, so it's still a challenge. And don't forget, you may have lifelong commitment to having to pay for these monthly injection of immunoglobulins and so on. Those are ongoing costs, right? What is the best construct, the best target? Still not quite there yet. Different types of T cells, are some of the open questions. How to make it lifelong, because some people will lose its effectiveness after a number of years, and we still don't know what actually determined how to make it lifelong. How to control better the toxicity, the side effects. And also the engineering facility that's needed could be a challenge in some countries as well. So in conclusion, just some of the commonly asked questions, the FAQ, is this an exciting potential new treatment for myeloma? Yes, for sure. And I think in the coming years, we'll see even better and better product, better and better targets. So big hope for myeloma patients for sure. Is this approved for myeloma treatment now? No. No doesn't mean that you cannot have CAR-T treatment, but you need to know that it's not approved. So when you go and have CAR-T treatment in China or other places, make sure that it is in the context of a clinical trial. And when it's in the context of a clinical trial, these CAR-T treatments should be free. Okay, so you don't go to somewhere in, I don't know, like Timbuktu, and they say, oh, we have a cure for this. We'll charge you 100,000, and it's already 80% cheaper than what you will pay for the US product, right? And then you go there and you think that it's a great thing. You pay 100,000 and it's actually not an approved drug. It's an experimental treatment, okay? Then you know, there's nothing wrong with going for trials and experimental treatment because it's important aspects of getting new treatment and so on. But in those settings, the treatment must be free. There must be a consent form. They must clearly highlight to you what are the risks and potential benefit, and you must make an informed choice. It's not going to the market and buy a vegetable, that kind of transaction, okay? So I think that's what it means. Could this be a cure for myeloma? We don't know yet, because cure really does need longer-term follow-up. Is this treatment safe? Generally, yes, but there may be some serious side effects that is much more serious than the kind of side effects that myeloma patients are normally used to seeing, you know, with our daratumumab, carfilzomib, Velcade, very different type of side effects. So you need to be aware of that, but they are, again, generally manageable if the doctors recognize them. What if I want to try this treatment now? Only available through clinical trial. Okay, by all means join. I personally have a patient from South Africa, interestingly, but live in Bali, so came to see me. And he said that I went to China for the legend, the last trial that I say, trial, right? And he went there and he had his cells already harvested. But he said that before I can get my cells back, they stopped the study, probably during the time that Johnson & Johnson was acquiring the company. So everything put on hold. So he said that I need, you know, so he said that I need you to give me some treatment to kind of hold my disease, you know, because I you know, and end up he used daratumumab. So J&J benefit both ways. But anyway, so daratumumab hold him, but it actually only give him a response that is a bit like PR. So he still have quite a lot of disease, but at least we're holding him, he's quite well. Then one day he come and say, hey, great, you know, the China guys wrote to me again and say, I'm ready to go back for trial, right? So he went back for the trial, he got the CAR-T, and his response was very good. You know, he went into complete remission. So it's true, you know, although sometimes we worry about what is going on in China, but, you know, he got the treatment and he went to CR, and he was very happy to share his experience with me. But this is all in the context of a properly supervised clinical trial with proper informed consent and so on, right? So I think, you know, I encourage people to think about this, particularly when you come to a stage of your treatment where, you know, the treatment options are becoming a bit limited. Do explore these possibilities. You know, in my setting, I do know people that are doing trials in the US and in China. I can make referrals. But I would also say that these usually are hard to get in because there's a queue of people wanting to go into the trials. And the nature of the trials is that, you know, they are limited to certain numbers of people only. So it's not everyone who wants to go have. So there are some of these limitations. And certainly in Singapore, we're looking to opening some of our own trials so that our own patients can have access to these kind of treatment a bit easier rather than having to travel out of the country. So I think I probably, you know, I was complaining that people are taking a lot of time. I probably took up too much time as well. So thank you very much for your attention. Yeah.
Thank you very much for the kind introduction. It's always a delight to come back to SGH because I'm an old boy of SGH, and barely 10 years ago, I still recognize some faces, some familiar faces, some of you who were treated by me 10 years ago. And it attests to the fact that right now you're looking so good, and it's like you just recovered from a bout of multiple myeloma. It attests to the fact that myeloma right now is seeing better and better prospects, and patients are living longer and longer. So I am standing between you and lunch, so I promise to deliver this in a very light-hearted manner so that you will get to enjoy your lunch shortly after this. So you have heard all the advances in multiple myeloma from the earlier speakers. We've indeed come a long way from the very first case of myeloma that was described in 1844, a very poor lady, Sarah Newsbury. She had no Velcade, no Zomita. As a result, she died a horrible death. That was how she looked like before her death. Her bones were all so contorted. But of course, this comes as no surprise because during those times, she was treated only with rhubarb, orange peel leeches. All right, so in those days, median survivor of myeloma, probably you're looking at 1 or 2 years. With the World War came the discovery of chemotherapy, and for a while in the 70s, 80s, 90s, we were relying heavily on chemotherapy to treat multiple myeloma. So when there's a limit to how much the body can tolerate chemotherapy, that was when we started doing autologous stem cell transplantation. Many of you have undergone stem cell transplantation where we take out some of your stem cells, infuse very high dose chemotherapy, and then infuse your stem cells back to the system. And for some of you, another way of transplant is called allergenic transplant. This is now rarely done, only for selected cases. Common to chemotherapy and stem cell transplantation is that these drugs used for these modalities are what we call conventional cytotoxic chemotherapy. They kill all dividing cells. As a result, you get a lot of collateral damages, nausea, vomiting, diarrhea, fever, and a very feared second malignancy, so second cancers many years down the road. So the discovery of the proteosome in 2000, at the turn of the century, I mean, this really is a major milestone for multiple myeloma because the proteosome was discovered, and those 3 chaps discovered proteosome won the Nobel Prize in chemistry. So proteosome essentially is the garbage bin of the cell. If I can put a lid on the garbage bin in the form of a drug called Velcade, then I can poison the cell with a lot of garbage, and this is how we could kill myeloma cells very effectively. So I'm sure all myeloma patients now know what is Velcade and had experience with receiving Proteosome or Velcade. So of course, with other drugs, what we call the immunomodulatory drugs, thalidomide and lenalidomide, suddenly we realized that, hey, we are actually converting myeloma from a terminal disease to a chronic illness akin to perhaps like hypertension or heart disease or diabetes mellitus. And of course, over the past 2-3 years, the next generation proteasome inhibitor like carfilzomib, pomalidomide, the newer drugs, ixazomib, alatuzumab, and daratumumab, which you heard earlier on from the earlier speaker, Dr. Molly. With these drugs in our armamentarium, we are secretly optimistic that with a careful and intelligent combination of these drugs, we could perhaps be looking at the cure of multiple myeloma. So right now, we target the internal signaling in the myeloma cells with drugs like lenalidomide, Velcade, and also we are able to target surface antigens of the cell in the form of immunotherapy. So daratumumab herald the era of immunotherapy for multiple myeloma. And it is indeed very pleasing that any one given time, more and more patients with myeloma are alive because of combination of drugs. Some chemotherapy, a lot of targeted drugs, and immunotherapy. However, this comes with a caveat. Although these targeted drugs do not come with the toxicity that we see with conventional chemotherapy, it comes with another form of toxicity called financial toxicity. So patients will find that the financial burden to pay for these drugs can be really, really tough. So take, for instance, right now with all these trials, all these drugs, in the Western guidelines, normally in Singapore and Asia, we follow guidelines from Western literature. I mean, what the doctor associations in America recommend, we tend to embrace that like gospel truth. However, in the Western guidelines, they advocate these very expensive drugs up front, but some of these guidelines do not show us the price tag. For instance, right now, the ideal way to start, or the optimal best way to start the patient with myeloma treatment is with VRD regimen, Velcade, Revlimid, and Dexamethasone. That could set you back easily by about $15,000. And if you look at the average GDP per capita spend on healthcare in these Asian countries, you see that you're looking at a few $100, and suddenly there is a disconnect between what is being preached in the ideal world and what we really can practice in the real world. So take, for instance, a patient who is younger than 65, 70 years old. The standard treatment right now, as recommended by most guidelines, is VRD treatment as induction. You get 4 to 6 cycles, then you go for stem cell transplant, and then you go on what we call Revlimid maintenance for at least 2 years or even more. So if you look at the price tag for this treatment, which is actually standard treatment, After you have completed 2 years of Revlimid maintenance, you are already set back by $500,000 US. And mind you, myeloma is, at this point, we still cannot say that we can cure myeloma. This is just talking about the first stage of treatment of multiple myeloma. So, I mean, we must always conserve our resources for treatment of the first relapse, the second relapse, and subsequent relapses. So, that's why your myeloma doctors are all working very hard, and usually we do not tell you that you have got finite resources, dump everything in the frontline setting, because we want patients to have resources and means to treat subsequent relapses. So that is very, very important. And currently, with all the new drugs that you have heard, many of these new drugs are now recommended in the front line when the patient has relapsed or refractory disease. And these drugs are just getting more and more expensive. And some more, right now the recommendation is that with these new drugs in the relapse setting, we want these drugs to be in combination because we want synergism, and these drugs have to be given continuously. So this is a double whammy actually, especially for whoever who is funding treatment, because something like this would easily bankrupt the entire healthcare system, because myeloma is actually one of the most expensive diseases to treat. Just to give you a rough figure, in the US, myeloma constitutes about 1% of all cancers. However, the expenditure on drug costs is 17%. So it's like inflated 17 times, because myeloma, there's so much development in myeloma, so many drugs available that is good. Patients are living longer and longer, but the cost to treat myeloma is extremely exorbitant. And so if you look at what is recommended, combination chemotherapy, for treatment of a relapse, entry level, which is VRD, Velcade, Revlimid, Dexamethasone. In the US, it costs about $500,000 a year to take this therapy. And this is probably the cheapest if you look at other more innovative therapies that involve carfilzomib, daratumumab, so on and so forth. So for many of us in Singapore, really, to afford something like this, you must have, unless you have a very good integrated shield plan, otherwise, the doctors are actually working very hard to help fund these treatments, and also we're always trying to get whatever subvention we could so that patients will get the best care. To pay out of pocket for these drugs, unfortunately, you have to be crazy rich Asian, because no one will be able to afford such a bill to treat multiple myeloma. The truth is that in the world, if you look at patients getting upfront VRD, or patients who are able to get all the new drugs that have been shown in the guidelines, only these countries in red, America, the Nordic countries, Germany, Japan. In Singapore, only if you have a very good integrated shield plan. Otherwise, many of us throughout the world, we do not have the luxury of getting all these drugs whenever we need them. So even for bread and butter, Proteosome and lenalidomide, right now these are really bread and butter for multiple myeloma, because every myeloma patient will be exposed to these two drugs in the front line. Right? So even for many countries in the world, Asia for instance, in Mongolia, the Kazakhstan, Myanmar, Laos, you do not have Velcade, you do not have Revlimid actually. These patients are still relying on conventional chemotherapy. And North Korea, for instance, no Velcade, no lenalidomide, no Revlimid. So this could have been a Nobel Peace Prize worthy moment coming out of Singapore. American drug in exchange for denuclearization, but it wasn't meant to be, unfortunately. They have other agenda. But one major advancement, another major advance in treatment of myeloma is our ability to measure the amount of disease that a patient has. For a long time, we were always contended, we were resting on our laurels when a patient has complete remission, that means CR. So I'm quite sure your doctor will come to you and say, hey, your piece of paper says M-band 0. Congratulations, you are in a CR. So we all feel happy. But then with technology, right now, immunophenotyping, flow cytometry, next generation sequencing, these and PET scan, these are expensive technologies. To be honest, we are able to measure deeper responses. That means if you still have minute amounts of diseases, we can measure. So long as we can measure these amounts, these minute amounts of diseases, we want to be more ambitious here. We want to eradicate more myeloma cells. So as a result, this is a vicious cycle. The more we can measure, or the more sensitive the technology is, the more we want to eradicate the disease. And as a result, this is a vicious cycle that the cost of myeloma will just get higher and higher. This is inevitable. So if you look at the US statistics, you look at inflation in healthcare, US GDP, probably about 200%. But for cancer medical, this involves diagnostics, the inflation is about 600%. But if you look at cancer drug cost, the inflation is almost 1,000%. So this is indeed very, very remarkable. And all of us blame the US for why your Velcade, your Revlimid, your Carfilzomib, everything is so expensive, because these are originally developed in the US, because you have to think about the high costs involved in research and development, because no drugs will just come like that. You need to go through many phases of testing. And unfortunately, in the UK, it's got to do with Obamacare. The Medicare cannot negotiate for drug prices. So the pharmaceutical giants would dictate the prices in America, and in America, most patients are all well insured. And unfortunately, these prices get transmitted down to the rest of the world. So why your Velcade is so expensive, why your Carfil is so expensive, is because the prices is pegged to the prices in the US. And also the reimbursement system in the US encourages more expensive options. There's always a middle person where you get a cut, and then this inflates the cost even more. And US, although all drugs have got a patent, there are always means to evergreen a patent, such that these patents become longer and longer, and this does not allow the generics to come in. So that's why this explains why the cost of these myeloma drugs are so expensive. So take, for instance, over the past 10, 20 years or so, these are the cancer drugs that are being approved by the FDA in the US. So in the year 2004, a magic bullet called Gleevec, which is a drug for a disease called chronic myeloid leukemia. If you've got this disease, your survivor is 4 to 5 years. But if you can lay hands on this drug, your life expectancy is no different from the healthy population. So when this drug was approved, it was about $4,000 or $5,000. Right now, as the year goes, with every approval, the drugs are getting more and more expensive. So this cluster represents all the myeloma drugs that were approved probably about 4 or 5 years ago: Carfilzomib, pomalidomide, Dexamethasone, daratumumab. Right now, these approved drugs, they are averaging at about $15,000 to $20,000 a month. So this inflation is indeed quite scary for many of us. So last year we had Asia Myeloma Network meeting in Beijing. We look at the member countries in Asia and see how these new drugs are available in these respective countries. So, well, if you have myeloma, the best place to be is in Japan, because it's all paid by the state. They have a national healthcare system, and all these expensive drugs are reimbursed. But in Singapore, I mean, to a large extent, whenever a drug is first introduced, most patients may still have to self-pay or rely on their insurance. So there isn't a perfect system in most parts of Asia. So doctors are actually working very hard behind the scene if you're a patient in a restructured hospital, because we are working with the authorities, MAF, trying to bring these drugs, make these drugs available to patients who need it. So there is a dire need to reduce the price of drugs. This is a global problem. It's as bad as global warming. We need to do something about it. So of course, the health authority need to do something. Health authorities need to negotiate for drug prices and control the formulary. However, with every negotiation, the risk is that the drug gets introduced later and later. This is unfortunate. And the health authority must facilitate earlier entry of generics or biosimilars, because generics are cost only a fraction of what you would expect to pay for compared to the original drug. So it's been shown that with more generic companies coming in, actually the cost of a certain drug would definitely come down. So for now, Velcade, Revlimid, the patents are still on in Singapore, but this differs for different countries. So of course, as doctors, we would always want these drugs, the prices of these drugs to be lower and lower so that this will ease the burden of our patients. And our pharmaceutical partners, yes, they play a very, very important role because without these drugs, many of our myeloma patients will not be alive today. However, I mean, it would be good that if they can lower drug costs and also the cost of drug development, there ought to be some transparency with research and development so that if we know that company A spends certain amount of money on research and selling this drug at this profit margin, I mean, we'll be at least more reassured that the company is helping us sell this drug and making this drug available to our patients. And also, we're always looking forward to value-based pricing. That means the patient pays only if the drug works. If the drug doesn't work, the patient doesn't have to pay. But unfortunately, this only applies to the CAR-T for leukemia children. But most for adult patients, unfortunately, we don't have this kind of system, value-based pricing, where you pay only if the drug works. So for doctors, especially for myeloma key opinion leaders, we have a responsibility to improve treatment guidelines. If we blindly follow Western guidelines, we will bankrupt the patient, we will bankrupt the healthcare system. So also education is very important. So it is our role to educate the patient why they need to be adherent to certain protocols or regimens. Otherwise, I mean, the cost of relapse may actually be more expensive than the cost of the frontline treatment. Clinical trial is also a very, very important way for us to reduce drug expenditure, the cost of treatment for our patients. So our Asia Myeloma group has actually come out with our own guidelines based on resource availability. This is for Asian perspective, which is a little bit more down to earth and more practical in the Asia setting. So recently we have an update, a revision of this guideline for Asian physicians. And in this guideline, we are trying to paint a picture that although there are advances, sometimes we don't always have to go for the Rolls Royce or the Ferraris. Sometimes Toyota may do just a good job. And also in this review, we engage doctors, key opinion leaders from India. Why India? Is because if you look at drug costs, for instance, in Velcade or Proteosome, this green bar represents American cost for Velcade, whereas India is here, I mean, probably like $100-200. Similarly for lenalidomide, American price is $10,000 US, but for India it's again a few $100s. So this is important, especially since the patent for Proteosome and for lenalidomide are coming to the tail end. And these drugs are very, very important so that we want these drugs to be combined with other drugs as well. So very important for us to know how to bring these prices down. So how about clinical trial? You've heard a lot from Prof. Chung earlier on. Will clinical trial benefit us? Certainly. So all the drugs that you have heard, all the novel drugs, by the time they move from bench to bedside, it will take at least 5 to 10 years. But patients are dying. So sometimes the best way to gain access to a certain drug is to enroll into a clinical trial. And the good thing is that in a clinical trial, the drugs are free. There's no free lunch in the world, but sometimes I tell patients that if clinical trial, everything is free. And so that is good. On the other hand, as doctors, we are always mindful that in the clinical trial, it is always a learning curve. Sometimes when we're dealing with new drugs, we are faced with unexpected side effects. For instance, like 1 week ago, FDA issued a warning on the drug called venetoclax in multiple myeloma. We were very optimistic that this drug could benefit a subgroup of patients with multiple myeloma. It came as a surprise to me that, hey, this was associated with higher toxicities. But whatever it is, I mean, as doctors, we are always keeping abreast with the latest in development so that we know the risks involved with clinical trials, with every single drug out there, so that whenever there are hints or signs that a certain drug may not be safe, we will always discuss with the patients. So in a nutshell, how we approach myeloma in this current era is that when a patient is diagnosed with myeloma, unfortunately, we always need to look at the reimbursement because myeloma is not just a one-time treatment. We're looking at the whole career, the whole is longitudinal. so that we know where to disperse funds when needed. So with modern day diagnostics, the doctor will decide what's the best way to treat a patient, whether the patient needs conventional chemo, transplant, novel drugs, whether clinical trial would be ideal for the patient, and in the tail end, if the patient needs CAR T, then we will need to see which are the centers that are doing CAR T and refer patients. And also, of course, at the back end, sometimes if a patient desperately needs generics, then there are actually resources for that. So whatever it is, I mean, in Singapore, no one should be denied of effective treatment to achieve the best outcomes. There are always ways to go around. So for a doctor, we are essentially your lobang king, so to speak. So lastly, do not underestimate the power of the patient. Right? So patient care organizations are very, very important. Patient support and education is very, very important. So I'm always inspired by this movie called Dying to Survive. Has anyone seen this movie? This is actually the summer blockbuster in China last year. So it loosely depicts a real-life story of a man called Lu Yong. He was a drugstore owner. He was down with chronic myeloid leukemia. That's the disease which I mentioned. If you have access to a pill, you survive indefinitely. If you do not, you survive 4 to 5 years. So at that time when he had the disease, he could not afford the original drug called Gleevec, which costs about $4,000 a month, and you have to take it forever. So he turned to India and obtained generics. In the process, because he was a drugstore keeper, so in the process he imported Gleevec for 1,000 other Chinese patients. And when the authorities found out, he was jailed for bringing in counterfeit drugs. But then again, this led to a petition, and eventually he was acquitted, he was released. And to us, I mean, he's not a criminal, but he's more like a folk hero because he's saved 1,000 other patients. So right now, because of this story, which is actually a real story, it has resulted in the Chinese government loosening up their grip on drug control. So right now, they're easing the availability of generic drugs because we need to save lives after all. So there is a fine balance to be achieved between respecting patents and intellectual property, because our industry partners pump in a lot of money to do research to improve survival of patients. We have to respect that, and charging a premium for certain drug, I mean, that is to be expected. On the other hand, we need to balance that with respecting lives. So this is represented by India, where in 1970 the India Patency Act did away with patents, such that in India you can produce generic drugs. As a result, the generic drugs are so cheap. I mean, many of us will know that many patients will go to India to buy generic drugs because it costs just a fraction of what you would expect in most the rest of the world. So a balance has to be struck somewhere in between. And also I want to take this opportunity to highlight the importance of patient advocacy. So recently there was a study called Make Blood Cancer Visible. This was a project done in Asia to really gather information on the public's perception of what blood cancer is. As you know, myeloma is a rare disease, it's a blood cancer, it's a very rare blood cancer. So if you were to speak to your friends or relatives, sometimes they will probably ask you, hey, what is myeloma? And there's a lot of misconception. I mean, linking blood cancer to death is something incurable, it could be contagious. And whether you're in Japan, you're in China, there's a lot of misconceptions about what blood cancer is. And in developing countries, as a result of this misconception or unawareness of what blood cancers are, this led to very late diagnosis for many patients with multiple myeloma with detrimental outcomes. So it is very, very important for the public to know what blood cancer is, what multiple myeloma is, and also for the patients, it's also very, very important for patients to know that you are not alone. So something like this is very useful because it gathers all myeloma patients in one single room. You know you're not alone. There are also other multiple myeloma sufferers like you. So by having unity or strength, we could actually do a lot. So recently, this is a study done by an NGO called Rare Cancers Australia. So as you know, Australia, being a typical Western country, their patient support organizations are very strong. They know what to do, how to grill the government. So they surveyed the various patient support organizations in Asia and look at how they go about doing the things. So, I mean, in the interest of time, I'm just going to summarize that. What they found is that most patient support group in Asia is more like a social gathering. Patient support group does not engage the government, does not engage the industry or the pharma companies. But these are very important because whereas in Australia, in the Western societies, the patient support groups are typically very vocal, right? So they know how to engage the government, the industry, and this is very important because right now we're facing a crisis. The treatment of myeloma, the drug costs associated with treatment of myeloma is indeed a crisis, and we need all stakeholders to come in to make treatment feasible for all with multiple myeloma. So education is important, and we actually need to identify patient champions and leaders and motivated family members to actually champion programs in a patient support group, and not just like education event like this. Actually, we can move beyond just a gathering like this, and also regional meetings for best practice sharing. So as a matter of fact, this year in October, in conjunction with the Asia Myeloma Network, I'm involved with Asia Myeloma Patient Support Group Forum. So actually, I would invite many of you to attend. In fact, we want to send Singapore patients to this meeting to interact with patients from other countries in Asia, so that together you can learn from one another and see how everyone engages the industry, the pharmaceutical companies. So in essence, as doctors, there's so much we can do. We empower you, patients, with knowledge, with education. We teach you about clinical trials, how best to deal with myeloma. However, with this empowerment, I mean, we always want patients to be proactive, to engage the industry, to engage healthcare authority, because we have a common problem, common agenda is to reduce the price of myeloma treatment so that myeloma treatment will be available to all in time to come. So to summarize, prognosis of myeloma has improved significantly. The key to improve survival outcomes is actually the access to the latest novel therapeutics. Regional collaboration is important to optimize patient outcomes. Escalation of costs involved in myeloma care may not be sustainable for many healthcare systems. As it is, I think if MOH looks at the healthcare bill size of a typical myeloma patient, MOH will really freak out because it's definitely not cheap. Engaging various stakeholders is key to finding solutions for affordability and access. So before I end, I just want to share with you a quotation. With access to treatment, a patient is able to live now. With access to support and education, a patient is able to live well. And with capability building, a patient is able to live for the future. Thank you very much.
The question is, can we have a comparison of Zomita and denosumab after the clinical trial ended in January 2019? And comparison on the cost, the side effects, and also biphosphonate are recommended to stop after 24 months. If resurgence of lytic lesions occur, what is the maximum interval for use of biphosphonate? Monthly, 3 monthly, 4 monthly, or 6 monthly? I think the conclusion after the trial is the denosumab is not inferior to Zomita. So really, they are equally effective in preventing the skeletal-related event. And at the moment, my understanding is denosumab is a lot more expensive than Zomita. The price of Zomita has come down recently. Side effect-wise, the Zomita dosage got to be modified according to the kidney functions. Denosumab, as far as I know, there's no change in dose. Correct me if my colleagues is understanding. Yeah. Fracture risk, they can all potentially lead to this atypical fracture of the long bone. And, about long-term use, yes, they can be used long-term, but I think in general they are used for 2 years in the first instance, and if the disease is still active, we carry on the treatment. Just I mentioned earlier on that the biphosphonate tend to bind to the bone matrix, and the effect can last a bit longer. And my view is that the biphosphonate can be stopped for a bit longer, whereas because the denosumab is an antibody, the effect in the body lasts for only about 6 months or so, compared to the biphosphonate, which bind to the bone matrix. So this denosumab, for this, if we need to use them, restart using them again more often. For resurgence of the lytic lesions that occur, that would mean that the myeloma is active again. We will still carry on using, for my practice, I will carry on the treatment, either Zomita or palmitoronate, while patients on chemotherapy every monthly, according to the schedule of the chemotherapy. But again, it won't be more than 2 years.
I think the only thing to add, there was a question about duration, if I got it right. From trial evidence, we know that using, whether it is Zomita or denosumab, we tend to use them upfront from the time of diagnosis of myeloma for at least 2 years from there on. Beyond that, it's not very clear whether we should continue to use it indefinitely or stop at some point. Most people would stop at some point after that, and then when the relapse happens, we tend to restart them. It's not wrong to continue to use it, but maybe in a less frequent schedule, maybe once every 3 months or so. And that is usually widely accepted by experts.
Okay, I'm very glad that we have a number of questions coming through. I'll just, one of them is quite interesting with a new... is this question of allergenic transplant versus CAR-T treatment? Okay, so the question was, what are the pros and cons? Which one is better in your opinion? And what are the risks? And I think this would be interesting to hear from the panel because it's, somewhat controversial. I would only say that allogeneic transplant is a much more established treatment. People have been doing allogeneic transplant for years. And in fact, the outcomes are getting better and better because of better ability to control the treatment-related problems. So the survival and outcome of patients undergoing allogeneic transplant is much better now. And also we know because of the fact that allogeneic transplant has been done for so many years that truly patients can be cured by allogeneic transplants because we have patients who survive 20, 30, 40 years when they do that. But allogeneic transplant is traditionally only available to much younger patients, patients usually below 50 years of age. So it's not something that every patient can undergo. So that's for allo. CAR-T is very promising because of the high response rate and so on, but it is not without serious toxicity. So when you say which one is more, have more complications and so on, I would say that actually the nature of the complications are quite different. The CAR-T complications, a lot of them are acute, short-term, dangerous, but you have these long-term effect that requires maintenance treatment. Similarly, for allo transplant, you have early graft-versus-host disease, late graft-versus-host, so there are short and long-term problems that the nature is a bit different. So they both have these short and long-term problems, so they're probably about the same there. The problem with CAR-T is that we don't know what's the long-term outcome, and it's the accessibility. Like today, you want to do an allo transplant in Singapore, we can do it. But if you want a CAR-T treatment for myeloma, I would say we don't have one, right? So that is the main issue. In 2-3 years' time, when you have CAR-T approved or trials available in Singapore, now these questions then become a bit more challenging, and I suspect people will be veering towards trying the CAR-T first before doing the allo transplant. I mean, that's my personal thought, but I would like to hear the rest of the panel.
I think most of the points that Professor makes are absolutely spot on. The thing to mention is most trials that have involved CAR-T, the patients did not go through an allergenic transplant beforehand. And second, it is even with the CAR-T, it's not one treatment, right? As Prof. Chung's session was he was mentioning in his session, there are CAR-Ts where you can harvest most of the time your own T cells and then re-engineer them before infusion. But there are now increasingly CAR-T treatments using other donor T cells as well. So it's going to be an evolving landscape of treatment. I think there is going to be quite a while before we get a clear answer towards which one is better. And they have different spectrum of side effects, right from the word preparation, everything is quite different.
So just to add, so what my colleagues have mentioned, definitely I agree. But the context of when we want to consider CAR-T, when we want to consider allogenic transplant, sometimes could be very different for a very, very young patient who may potentially wants to be have a chance to get the to be cured of multiple myeloma, and he has got bad myeloma genes. Then allogenic transplant is something that I would consider. On the other hand, we also ought to look at the reimbursement. So right now in Singapore, if you're Singaporean, allogenic transplant is very heavily subsidized by the government. But if you're talking about CAR-T, we have absolutely no idea how much it's going to cost. But right now, as a ballpark in America, if you go for CAR-T, just the product of the CAR-T alone, minus the hospital stay and so on, you're looking at about $400,000. When it comes to Singapore, I do not know what the price is going to be. So finances is also another major consideration before we decide what is best for the patient. But things are still evolving, and of course, we'll be more than happy to just to keep abreast with the latest in the literature, so that when the time comes and when you need to make the decision, then we all the doctors will give you a very unbiased opinion on what is best for the individual patient.
There's another question about stem cell transplant, because it was mentioned that nobody mentioned anything about stem cell transplant. Doesn't mean that it's not needed now. And the question is, what's the difference between that and the CAR-T? I think there are many things that we didn't mention today because they are standard already. I mean, nobody say much about Velcade and so on. So these are standard treatment that all myeloma patients will go through, including the stem cell transplant. It is definitely still beneficial, and like I said, the CAR-T is something that is not available now. It's something for the future. Now, certainly in the future, it is quite possible that if the CAR-T is mature enough and really safe enough and effective enough, that we may not need to do an autologous stem cell transplant. That's quite possible, but not by today's standard. Today, the autologous stem cell transplant in Singapore is relatively cheap, is very safe, is very effective. It's heavily subsidized, as Daryl have said. So cost effectiveness is really very outstanding. So I think we'll do that. The CAR-T treatment is, in terms of procedure of what we do to the patient, is about the same in principle, but actually behind the scenes, the science and the thing is actually completely different. The principle or the concept of treatment is actually quite different. The autologous stem cell transplant is using the chemotherapy to treat the cancer, which is the high-dose melphalan. The stem cells are to rescue and allow the body to recover. Whereas the CAR-T is completely different. They are using the immune cells to attack the cancer. So CAR-T in principle is closer to allogeneic transplant because the allogeneic transplant principle is the same. They're using someone's donated immune cells to attack the cancer. So the allogeneic transplant is actually the original old version of CAR-T, if you want to think about it in that way. So they're quite different actually, but today stem cell transplant is still, autologous stem cell transplant is still one of the key standard treatment that we have. Any other comments?
Nothing much really. Just wanted to reiterate that, yes, absolutely, transplant is useful in the upfront treatment of myeloma. Sometimes if we've been lucky with a patient that one transplant gave them a fairly long period of remission, either with maintenance or without, and if the patient is still fit second time round, well beyond 3, 4, 5 years, if they managed to remain very well with myeloma, we do consider a second transplant as well if they are fit at that point. So, and in our context in Southeast Asia, really, if you think about the kind of treatments that are available, stem cell transplant may not sound that cheap, but it's actually one of the cheapest ones that we have. So I think from both what Darryl, Dr. Tan was mentioning about the cost of treatment, it's relatively cheap and safe, not that we have been doing it for years and years and years, and do consider transplant as a very important part of the treatment for myeloma.
I'm from Tan Tock Seng. We do not do transplant in Tan Tock Seng, but we refer our patients who need transplant either to NUH or to SGH. But I do notice between the two hospitals, the criteria for transplant sometimes may be a bit different, in particular for patient who has got renal impairment. And you see the patient who has got renal impairment, the toxicity of the procedure is higher. And so there's even within this, there are two different views between the two hospitals. That's my observation.
Just move on to some other question. There's one relatively simple one that I'll just answer first. So someone asked about average time for remission of myeloma. In myeloma, as you know, the patients usually have the disease, get treatment, get better, so in remission, and then relapse again, treat, remission again. So there are actually multiple times of remission, but very often the remission is the longest the first time, and then it usually get a bit shorter and a bit shorter and a bit shorter subsequently. On average, the first remission, using our data and international data, is usually about 3 years, but with newer drugs introduced to the beginning, this time may keep changing in the future. But it's very variable, this average time, which also goes to show that some patients may relapse very early. We certainly have seen patients relapse after 2-3 months of the transplant, within the first year, but we also have patients that kind of relapse after 7-8 years, or some actually never relapse, right? So it's a big range, and the usual advice to the patient is to just continue to follow the instructions of the doctor, go back for regular checkup so that we can pick things up early, and don't obsess about numbers like this, because it's really different for a different patient, and you will not know for yourself what it is until it actually happen, right? I mean, so it's these are the general pattern. Mauli and Darryl, want to add anything to that? No? Okay. The next question is on maintenance therapy. This one is, as usual, quite controversial. So the questioner was asking panel's views on maintenance drugs in terms of efficacy and financial burden. Maybe Darryl, since you're a financial person.
So let's say, for instance, a patient who has come off transplant, and usually we will discuss about maintenance. So this discussion actually I take into account many factors, because your reimbursement, because right now there are a few ways we can do maintenance. One, the standard or what is recommended is with Revlimid. But the cost of Revlimid, if you apply it in an indefinite duration, the recommendation right now is that if you're on maintenance, you do it indefinitely, because you are as good as you are on the drug. Right? So this is what the latest studies have shown. With Revlimid, the cost is the issue. So if a patient is well insured with integrated shield plan, yes, that is ideal. Anyway, that is that's recommended in all international guidelines. However, if a patient has got financial difficulty with Revlimid, then the next consideration is a drug called thalidomide. But unfortunately, thalidomide is the first generation lenalidomide. It comes with this side effect called peripheral neuropathy. Sometimes it can be very troubling for the patient because of numbness and tingling sensation on the feet, and sometimes it could be permanent. So as far as possible, if financially it's feasible, I do lenalidomide first. If not, then thalidomide. But thalidomide usually you can never do indefinitely because many patients will have side effects. And on the other hand, I also look at the genetics of the disease. If a patient has got bad genetics, like 414 translocation or deletion 17p, so these are bad genes, it has also been shown that with lenalidomide alone, it may not work. So these are the patients where I may consider doing Proteosome or Velcade as maintenance. When we're talking about maintenance, we're talking about taking a drug like it is your anti-cholesterol medication long-term. So long-term, the side effect is always a consideration. Again, with Proteosome, long-term side effect is still nerve injuries. Now there's a new drug called Dexamethasone that has been tested. It's like an oral form of Velcade, but you take the pill once a week as a maintenance. So for patients who already have got numbness or nerve injury, Dexamethasone is a drug that could be considered, but then again, this is another very expensive drug. So overall, I don't think there is a standard answer to how anyone does maintenance. I look at several things: patient factor, patient's reimbursement, and patient's genetics and the risk of the myeloma before putting forth a recommendation for the patient.
Anything to add, Mark? The rest? Nope.
I completely agree. Sometimes with the management of myeloma, it is almost nearly like solving a Sudoku puzzle. So you just put the different pieces, the patient, the genes, the myeloma presentation, the patient's toxicities that they've had from treatment, from the disease, and then you put everything together, then you get a neat puzzle that we know how we solve this best. So I think there's no one consideration for every patient. It's just we're completely individual dependent. I don't have anything to specifically add.
So on the other hand, I have got some patients coming from Indonesia. And they actually already sold their house to go for a stem cell transplant. So these are the patients. Sometimes I would say maybe instead of selling another house to pay for maintenance, perhaps you could consider conserving your resources for the treatment of the next relapse and don't do maintenance. So that is also a possibility just for patients who are, especially patients who are self-paying and getting a little bit tight financially.
So while the mind is with Mauli, maybe I'll ask him a question. There's this question that was asked. It's not quite complete. I hope I get the question correct. I suspect this is in relation to the new drug that was probably your media release on carfilzomib, I suspect, because it's directed to you and there's a new thing. I think so. I'm not sure who asked that question. Is this correct? You're interested to know about the new drug that was mentioned in the newspaper for myeloma?
Yeah, it's me.
Yeah, oh yeah, that's the one, right? Yeah. Yeah.
The trial that was mentioned actually was new at the time of launching. The drug was new at the time of launching. It's not new anymore. Many of you might have known about the drug called carfilzomib, which is now in Singapore, been around for about 5 to 6 years now. So that was the one that was, it was mentioned. Was there a specific question about the drug?
No. What is the outcome and progress for myeloma? So maybe a bit about the result.
Okay. So that particular trial involved, so now we talked briefly about the phases of treatment for myeloma. So the first phase, as soon as patients get diagnosed, is what we call as induction treatment. And for some time now, we know that when we use a combination of 3 drugs, patients do better, the outcomes are better than compared to using just 2 drugs. And so that phase of induction, we used 3 combinations. Usually Velcade was one of the drugs, either cyclophosphamide or thalidomide being the second drug, and dexamethasone being the third drug. So in this particular trial, we replaced the Velcade with carfilzomib. So we used carfilzomib, cyclophosphamide, and dexamethasone as an induction treatment. This was a trial that was specifically used for patients who were eligible to have a transplant. So after a certain number of cycles of these 3 drugs, patients went through harvesting of their stem cells, and then they went on to have a transplant. After they finished the transplant, they received 2 more cycles of the same 3-drug combination of treatment. This phase of disease treatment is what we call as consolidation treatment. Then we go on to the maintenance that we were all talking about. But before they went on to a maintenance phase, patients had a bone marrow assessment done, and they had something called the minimal residual disease assessment, which Dr. Darrell Tan was talking about earlier. So in patients who had still detectable amount of disease, but in a very, very small quantity, that is what we call as MRD, patients went on to have this maintenance treatment with the same drug called carfilzomib, which was all, obviously this is not part of a clinical trial, and hence the medicine was completely free for patients. And then subsequently, we continued to assess the patient, and we now have reached a stage that we were able to look at analysis and see how the patients have done. The other important thing about this particular trial was this was for what we call as high-risk myeloma patients alone, which means that patients who had these very expected to do poorly with really bad kind of myeloma genes within them were the patients whom we recruited into this trial between SGH and NUH. So we look at and compare. So this was, within the context of Singapore, we know the population is going to be small. So we didn't have any comparative arm for what we use this. For example, we could have compared saying, okay, we are using carfilzomib with this. Why didn't we compare it to Velcade? But if we have to do that, it will be much, much more cost-wise for running these trials. But also, how do we assess? So then we have to compare it as such, as a single-arm trial where how much have these patients responded. So we had very good response. Nearly 60 to 70% of patients reached what we call as a complete remission, which we know is good, but not the best. But we do know that nearly 25 to 30% of patients reached a stage where not only were they in CR, but they actually reached a stage where even when we use special techniques like MRD, we couldn't find any evidence of disease. So it's still an ongoing process. We're still collecting data so that we see, how on a longer term, beyond 3, 4, 5 years, the patients who are particularly become minimal residual disease negative state, how do they do? Because it's important that in these high-risk patients, how they perform when they get to that stage. It's important for us to find out how it goes for them.
Yeah, maybe I would just add and make some generalization that, although we have improved a lot in myeloma treatment, there's still some small group of patients which have, bad disease, what we say high-risk disease. And that is the group that we still need to work very hard to tackle, to improve and make them as good as the rest, right? And the generalization is that all these new drugs that we're now using to treat relapsed disease like daratumumab, carfilzomib, if they are good in relapsed disease, they are probably better earlier on in newly diagnosed. So as we move these treatment earlier and earlier on, there is a cost issue for sure, but actually the outcome of the patients will probably continue to improve, which is why CAR-T now we saw treating patients with 3 or 4 previous relapse. I would say that if it's a successful treatment in the future, it could be CAR-T right up front, and it will potentially change the landscape of these patients, because if you don't relapse, you're cured at the beginning, then all the costs that we're talking about may be more compelling to even pay more upfront if you're cured, right? Rather than keep paying, over the years. So I think this is the trend that things are moving, and in the coming years, you'll hear more and more of these things. I want to touch on this one, which I think is also important. There's someone who asked a question about dexamethasone, right? Patients are surviving way longer now. There's many rounds of treatment. Every round you get dexamethasone. You seem to be on dexamethasone all your life. And there's a lot of side effects. The cataracts start to come, the infections start to come, your legs are swollen, you're getting, diabetic, and so on. The question is dexamethasone necessary in all the cocktails? And what if you don't use it? What would be the downside? And maybe linking, everyone can answer this question.
Yes. I do notice a patient who've been on dexamethasone, they may they develop cataract as well as fracture of the hip due to osteoporosis. My practice now is, I try to cut back on this when I see patient has a lot of bone disease to begin with. For instance, if my patient's on lenalidomide maintenance, I will withdraw the dexamethasone weekly after some months. If the patients manage to keep the disease in check, I will stop the dexamethasone part.
I think I would completely agree. I do fairly same thing. When patients, it depends on patient's response, right? I mean, induction treatment at least is finite. You get 4, 5, 6 cycles of treatment, and hence the dexamethasone also stops at the time of induction treatment ending. But it's usually the maintenance part that is particularly a problem. So you start off with lenalidomide and dexamethasone, and then there's no stop date to it, unfortunately. However, if you're cognizant about how the patient is responding, if the patient has already reached a stage of very good remission or even a complete remission, is there a point really of continuing the dexamethasone? Probably not. Probably not a strong case in any way. So we do think about that, those patients. And secondly, I go back to the same issue. It's not a one-size-fits-all approach. It has to be based on the patient's response, and especially with regards to the toxicities. We use the same dose of dexamethasone with the same combination of drugs. One patient has absolutely no issues whatsoever, but the next patient, is absolutely wrecked because of dexamethasone. They have, cataracts. They become diabetic. They weren't diabetic before they started. So we have to teeter it to how the patient's tolerability to that particular drug is, not just for dexamethasone, but for everything else as well.
So if part of your question is whether we have evolved to such an extent where we can drop dexamethasone, unfortunately, the answer is still no. I mean, we celebrated when we could do without conventional chemo up front. And for the longest time, we were using this chemo called VAD. It's like leukemia chemo where a patient stays in hospital, you need an infusion up front. So when we stopped that totally, we were quite elated. But dexamethasone, unfortunately, it is still essential, especially in the beginning, because it's an anti-inflammatory drug. So in the beginning, when patients come to with myeloma, there's some inflammation here, we need to quickly arrest certain things like high calcium. So dexamethasone in the early phase of myeloma treatment is essential, but later on, depending on the side effects, we will want to tail it off. And sometimes dexamethasone is actually a very good drug in combination with other drugs. If you're let's say if you're on Dexamethasone, you can sometimes get very bad nausea, diarrhea. The dexamethasone is the one that is actually suppressing all these side effects of some of these drugs. Yes, we are still evolving right now. The dose of dexamethasone, we have found that we don't need high dose, it's cutting down. But then again, right now at this point, especially in the front line, or when we combine with chemo, other new chemos, it is still an essential drug, but we are all mindful that there are toxicities, and monitoring for side effects is always at the top of our priority.
Yeah, so I would just add that what my colleagues say are all true, but we also have seen that over the years, as Daryl has said, we've gone from chemo to no chemo. In fact, in the past, we used to give a lot more steroids, 4 times more steroids for treatment. And there was a study that shows that, in fact, the way that we give it out, which is once a week, is as good and, in fact, safer, less side effects than the higher doses of steroids. So I'm also I don't think it's beyond possibilities that in the future we can give less steroids. Whether we can do without steroids or not is a completely different question. And I think within the context of the Asian Myeloma Network, we're quite keen to look at some of these studies that are not very sexy new drugs type study, but may have real impact on patients' outcomes. So one of the studies that we are thinking of is actually to look at comparing a 40 milligrams per week with maybe a 20 milligrams per week dexamethasone with the rest the same. And there is no rhyme or reason why 40 milligrams is, it's not based on any very good scientific rationale and so on. So, why not 20? And I think these are some questions that we can ask. And I think if we can see that it doesn't impact on the response, but may benefit the patients, then we can, slowly see whether we can get away with less and less steroids that will help our patients. So I don't know about time. Are we? 哇，还有的。 Zidian, when's the finishing? Just keep going for the whole day, is it? Okay. I mean, I would, let me see how I went on. Okay, let's do this. So there's one question on, this questioner asks, I've heard about whole body hyperthermia combined with chemotherapy in moderate doses to treat myeloma. What's your opinion? My opinion is I've not heard of this, so it cannot be standard of care. There are many, if you go onto the web and you read all the blogs and so on, people have tried different things. We also know that there are a lot of, I don't know, whether they are practitioners or not really qualified licensed practitioners, but the people doing all sorts of funny things out there, just like using stem cell therapy to make you look younger and all those. Those are non-approved treatment, right? Whether they work or not is another thing. But all I can say that is not approved. It may work. I'm not saying that it will not work, but it's just that it hasn't been robustly studied and certainly not treatment that we will recommend you to try, because there are other much more established, much more effective, much more well-established treatment that we can give. And I think, this is one example. There could be another 20, 100, 200 examples of funny treatment that people may have said that it will work, because whatever you read on the web would be things that have worked. Nobody will tell you things that have not worked, particularly if that blog is from someone that is an advocate of that thing, right? So I just caution, always check in with your doctor before going to try these things. Any other kind of advice from our panelists? No, haven't heard of this? Daryl? No? Linking? No, yeah. So absolutely haven't heard it before. The next one on diet during remission. Okay, when someone has finished the treatment and is well and so remission, any special dietary recommendation? Just first disclaimer is there's no dietitian here. But anyway, does anyone want to give some thoughts on this?
I guess it again depends on what stage of remission you're on, whether you're on maintenance treatment or not. If you're on good going amounts of lenalidomide, you might still have periods where your neutrophil counts are going to be low, and hence you still need to watch what you eat. Second, it also has to do with what have you been left with in terms of complications from before. For example, some patients post transplant can still be left, even when you're on no treatment subsequently, you can still be left with some kind of borderline blood count. So I think the bottom line rule that I tell my patients is healthy, hygienic, properly cooked diet. Unfortunately, no dodgy takeaways and no kopitiam for most of the patients, especially around the transplant and the peri-transplant period. Other than that, I think there are lots of concerns about supplements and things, and I think it was not last year, the year before.
Yeah, there was 1 year
when we had a dietitian. I think it's maybe a gentle reminder for us to probably think about the next session to include a dietitian again, maybe.
So I get this a lot. There's always a myth that you've got cancer, you should starve your body of glucose and sugar. So no, we do not believe in this. In fact, I have one patient, an elderly lady with myeloma, so I gave her a very simple treatment, and 1 month later after diagnosis, she lost so much weight and she was so scrawny that she was going to collapse. She looked like she was going to collapse any time. When I found out it was actually his son who refused to let this lady eat, thinking that starch, sugar will feed the cancer cells. So actually that was detrimental to her health. So to be honest, I mean, myeloma is not just the drugs that are killing the cancer cells. Your own immunity is also fighting the myeloma. So do not starve your immunity. Even if the concept of no sugar is correct, you're starving your immunity. Your immunity is needed to fight myeloma as well. So have a normal diet. Do not deplete yourself of sugary or starchy food, because that myth of starving your cancer to death is probably unfounded.
Yes, I was just about to say the same thing. In fact, we need good nutrition for our good immune system, our strong body. And if we were to undergo chemotherapy, we need a strong body to be able to withstand all this chemo. It's very tough for anybody to receive chemo. These are all cytotoxic medicines. Every round of treatment, we are weakened by this. So are we going to get ourselves even more weak by starving ourselves? Of course not. We need to be ourselves strong, get good nutrition, do not believe in the old Chinese supposed way of selecting certain food and cut off certain foods. That's my comment. Of course, nutrition supplement-wise, we always prescribe calcium and vitamin D. Yeah, but be careful because we check the blood calcium level quite often. If the blood calcium level is too high, then of course you need to be stopped for a while. That's my comment.
And just also be cognizant of the fact that many people are going to have different other things associated with myeloma or even unrelated to myeloma. You might be diabetics where the diet is going to be different. You might have kidney problems where you might have been told to avoid specific foods that are rich in potassium and so on. So really, if you are in doubt, ask your doctor to refer yourself to a dietitian and take what is the right thing that is suited for you.
Yeah. So this all the advice here is absolutely correct. Very often, this question is asked, anything I cannot eat? Then usually, no lah, eat everything lah. Then the guy look at me, is like, what? So I can tell you that in the population, the public, right, it's this conception that if I'm sick, particularly because Chinese is very strong in this, cannot eat certain things lah, chicken cannot eat lah, seafood cannot eat lah, very poisonous lah, and all these things. It's actually not necessary. There's no real solid basis to this, is all I'm saying. So there's another thing about food here, not quite exactly, it's about Japanese seaweed. Would this interfere with treatment or help with treatment? And again, I will let the others answer as well, is that I don't know, right? Because again, these are not really scientifically studied and so on. So I really don't know whether it will help or not. I mean, a lot of patients do come and ask, can I take some herbal thing here and there, with the treatment? My usual advice is that when you are taking your active treatment, like your Velcade and so on, better not take to interfere. But when you're in remission and you're not taking any treatment, if you want to try some of these herbal things or whatever, it's okay to try, as long as ask your doctor, make sure it's not something that may be dangerous or controversial. If there's no harm, you can actually take, and then, if it helps, it helps. If it doesn't, then, it doesn't cause any harm, right? So it's a practical approach. But I don't know, I'm not familiar with the literature. Do you all know anything about seaweed?
I didn't imagine seaweed around the different parts of the world would be very different. So I thought Japanese seaweed is probably the same as the rest of the seaweeds, but apparently not.
Yeah, I don't know. So, Darryl, any knowledge on this? No.
Not Japanese seaweed, but Japanese green tea. I mean, there's some interference with the absorption of Velcade. So usually when you're on Velcade, I ask patients to avoid green tea, not seaweed.
Yeah, correct. So we don't know about seaweed as such. Linking also don't know. Yeah. Any other question? Yes. Baldev.
Thanks for the fantastic talks. A lot of Indians believe in turmeric as a very good supplement for anti-inflammatory. Benefits of turmeric.
Interestingly enough,
Is curcumin from turmeric?
yes, correct.
Yeah, so
That's the
curcumin,
compound.
Some of the people are curcumin.
Well, it's a very, very good and very relevant question. One of the foremost investigators in the world for myeloma is Dr. Vincent Rajkumar. As the name might give it away, he's actually an Indian born in South India, where there's a lot of curcumin and turmeric being used. And I believe he's studying the effect of myeloma, effect of turmeric on myeloma, but I don't think the results have come to being kind of used as a treatment on its own, but it's going to be something that's going to be used with other treatments. And the short answer to that is the results are not known yet, but when they used it in a Petri dish on myeloma cells, there's quite a robust evidence to say that the myeloma cells are being killed by the compound. So if it is going to be part of your diet anyway, I don't see why you shouldn't continue. But if you are talking about specific dosages in terms of tablets and stuff, I don't think there's any guidance to say how much you should take or so on. But there is definitely a clinical trial on curcumin in myeloma.
Yes, sir. Yes.
Fucoidan. And they say that some Japanese doctors have studied and say that it's effective. But when you say what I'm doing now for myself is that I take it, but outside 1 day before and 1 day after the Velcade treatment, I don't take it. Is that good enough? or I should avoid altogether?
Any? Oh, so no. The question is related to the Japanese seaweed, and the gentleman is saying that I don't know whether he read online or somewhere that the seaweed may help. So he's taking it, but he's asking that if it sounds like he's on Velcade treatment as well.
Yeah.
So if he's taking the seaweed but stopping the seaweed one day before the Velcade, and then also don't take one day after the Velcade but take on other days, is that okay? I guess a lot of these things is that, so first of all, truly we don't know whether it will help or not. Is number one, that's the one first issue. The second issue is that does it interact with the active drugs that we're giving you, which is Velcade? So the example of green tea, we know that it interacts, so you shouldn't drink green tea or take high vitamin C containing elements, because that's another thing that has actually been shown to interact with Velcade. So if you say that I stop 1 day earlier and then 1 day later, then the question is, we don't know how long the seaweed stay in your body, right? So if the seaweed stay in your body for 2 days or 3 days, then it will interfere even if you stop. So there is a lot of these questions that we cannot answer just because nobody has studied how long because after you eat the seaweed, it gets digested, there may be some chemical compounds that then get absorbed in the body. So we don't know how long these stays, and it may interfere with the drug absorption and so on. So these are the kind of questions that I guess we don't feel qualified to answer just because we don't know, and nobody has studied the seaweed in that kind of detail. So the usual advice, like I said, I will give my patient is don't take herbal medicine or seaweed when you're taking the active drug. Particularly if the drug is working well, just carry on with it. Why? But you can take these other things after you finish your treatment. Because some people say, I want to I want to adjust my thing. After that, make me feel better, adjust my well-being. Then you can take these things after the active treatment is finished. Because while things is working well, it's fine. But some people may come back with some odd side effects. Then we have to wonder, is this due to the drug that we're giving you? Is this due to the seaweed? Is this due to the interaction with the seaweed? Then we are in a bind, what to adjust, right? What to adjust. Because we know that side effects may happen. But a lot of patients come in with sometimes unusual side effects. Then upon asking, you realize that they're taking all these other things out there. Then really, maybe stop all those things first, then continue with the drug. If it's still there, then maybe do it. Then we have to play a lot of detective work, which may make it very difficult to manage. I mean, that's the advice. I'm sure the advice will be similar.
I think so. May not be directly related to the seaweed, but there are many marine organisms and plants that have got very robust anti-cancer activities. So we don't. I guess we don't really know. There is one recent myeloma drug that we came to know about called Dexamethasone. It's actually derived from one of the marine compounds, and it seems to have even more robust activity similar to Velcade in controlling myeloma cells. So what we don't know, I guess, it's not just simply dismissing things we don't know. It's just that if we truly don't know whether it is going to add on to the side effects of Velcade. If you have, I mean, it can be completely naive looking as a seaweed, but if it really has a compound that can either interact with or might very well be very helpful in controlling myeloma, but in that process, if it has a similar mechanism of action, they can actually add on to the side effects of the same drug that you're going to be on. So I guess unless we are, in this day and age, unless it is specifically studied, we unfortunately cannot give out a recommendation.
But can we treat it like we take any other normal food? Because if we take other normal food, we also we take a lot of other foods, which we are not quite sure whether they have any other compound.
Yeah, so if the seaweed is in the, I don't know what this seaweed is. If it's in the normal context of the seaweed we take with some of our food, then it's fine. But if it's a pill or a concentrate or a different kind of thing that is derived from the seaweed, then I would say caution, because the dosing may be quite different. It may be many times higher than what you will eat from a seaweed that you put maybe when you go to a Japanese restaurant and they put some seaweed in soup. Yeah, so be careful. Although it's from the same thing, but sometimes the concentration does matter as well.
Understand.
Yeah.
Yeah.
So same thing as what Mauli have said about turmeric and so on, right? If it's the usual in the diet, but if you buy a pill, a turmeric pill, curcumin, the dosing may be quite different, and that it's not controlled, then we don't know.
So if I take at a normal, like the Japanese people, they actually take this seaweed. So if I take at the same level as what they are taking per day, is probably in the normal diet, is it probably okay? Because actually, the first few weeks I do feel a bit uncomfort in my body. But when I take this Fucodan, it seems to help. I take it normally on the third day after my treatment. So I take it on Thursday, I have my Velcade. Friday, I don't take. I take it only on Saturday, then I start to take one pill, which is, they say, is equivalent to the normal diet for the Japanese who is taking this seaweed.
So again, the rest can chime in, but the advice here is that if it's working well, I mean, you're feeling well, there's no side effects, and when you go back to your doctor, they check your response is also going well, then there's no harm. But just be mindful that there may be harm. It's not always just benefit, right? So there may be harm. If you experience some unusual side effects, or your doctor is saying that, hey, your response is not as I would expect based on the other patients that I treat with myeloma on the same drug, then you are in a way taking an educated risk, then you must stop the seaweed or something after that. But if everything is going well, again, I think it's a practical approach. There's no, it should be fine.
Okay, thank you very much.
If you feel well, okay, the monitoring seems to respond well. In your case, if you feel well taking it and the treatment progress well, that's probably okay. But it may not apply to the rest of the patients. The rest of the patients, if they want to try, they got to be very cautious. Yeah, that's all I want to say.
Okay. Any other? That's it. I've been given the thumbs up that's it. Thank you very much to the panelists for the very active discussion. Thanks for all your questions. Thank you. Okay, time is moving on. I think we are coming to the end. I just want to give a short closing. I just want to remind people that this is the 9th year already. Next year is 10th anniversary. It's going to be a big bash somewhere in Orchard Road. No. We'll plan a nice event next year to commemorate 10 years. But I'm also wanting to just touch on a point that Daryl mentioned. The group has gone from really a social support group for many years, 9-10 years, and some people have become very close friends. But I think it's come to the level of maturity, and the myeloma treatment is really such a complex and very expensive area that I've been encouraging the group here to really consider more taking on some sort of advocacy role, to have more of a patient's voice. I recently, this week, attended a meeting that was organized by The Economist. It's called The War on Cancer. And there are many of these patient groups from the region and the world that come together to talk about advocacy, capacity building and so on, to really see how we can benefit patients with cancer. And, the key thing that come up is that we need people that have passion. We need credibility, so the group must organize themselves so that there is credibility. And I think over time, we have many, many potential credible people in this group that can potentially take on that role. So I will encourage the patient group to think about this, especially amongst people that are capable and interested, because I think increasingly with all these very good treatment, but very expensive treatment, the worst thing is to know that you have good treatment, but you cannot reach because they are all so expensive, right? That we need to see whether we can do more to help access for the patient or not. So thank you all again for coming, and we look forward to seeing all of you again next year. Thank you very much.